Right, I think we can resume this webinar. First one in 2022. I believe every one of you that is joined has been joined since the very beginning, so I'm not going to introduce the whole seminar again, but we are going to continue now with a talk by one of our more senior PIs at the center, Johan Elf. He's been with us since the very beginning, and he is going to talk to us about his project looking into antimicrobial susceptibility testing. And I can see, Johan, there you have a very beautiful image of bacteria behind you.
Oh, thank you.
I guess you're going to show many more on your
Yeah, those are
yeah,
those
great.
are E. coli.
So you can share your screen and then start your talk. Thank you so much, Johan, for being with us and updating us on how your projects are going.
Thank you. Can you see the screen?
Correct, yes.
Yeah, thank you. Yes, so thank you for having me here. I will talk about rapid antibiotic susceptibility testing. And I should, in addition to my affiliation with Uppsala University, I should also say that I've started this company, Astrego Diagnostics, and I think I will not say anything incorrect just because I have an affiliation with the industry, but I should still disclose that. The talk will be about how we can go from this plate antibiotic susceptibility testing to a rapid small machine that you can see to the right here. And I don't need to explain the importance of antibiotic resistance to this audience, but why do we want rapid antibiotic susceptibility tests, or ASTs? And I'm sure most of you are aware of that misuse of antibiotics is a major cause of antibiotic resistance. And these are two different things, really. So it's partly the use of antibiotics for non-bacterial infections, such as viral infections, for instance, or the misuse of broad-spectrum antibiotics just in case. So maybe the doctor thinks could be a resistant infection, and then you give something stronger that is really not needed and drive resistance development. And why is it like this? One part of the reason is that the current methods for susceptibility testing are too slow for guiding the primary treatment. So plating cultures takes days, and then the doctor wants to prescribe something directly to the patient, and then it's too late, right? If we would have a fast AST, it would make it possible for doctors to directly prescribe an effective antibiotic at the point of care. So ideally already at the primary care center. And the question then from a kind of academic standpoint when we started with this in 2016 was, is this even possible? And the guiding the timeline that we had to operate with here is like 30 minutes. So that's how much time that in the primary care that you can expect someone to wait before you need to give them a prescription. So is it even possible? That was the starting point for this this work, and we were then my lab is normally not working in this kind of applied methods or diagnostics, but we have been developing very accurate and sensitive methods to study processes inside cells. So looking at bacterial cells and measuring things inside with fluorescence microscopy, in this case looking at replication forks inside E. coli, or even in this case in tracking individual fluorescently labeled proteins as they move around inside the cells and get trajectories out of how they move. Some other examples we've been looking at fluorescent assays for binding of transcription factors to individual loci in the chromosomes, or this is an example of homologous recombination, so looking at DNA repair at the individual cell. So this is kind of the background where we had all these techniques and very sensitive measurement techniques and image analysis tools. And the question is, could we make something that could help us determine antibiotic susceptibility in like 30 minutes. The starting point for that was a microfluidic chip, which allows very fast loading of bacteria directly from a sample, and we were thinking about the urinary tract infection sample already at the beginning. So what you have here is a fluidic ship where you can flow the bacteria or the sample and then trap them in these microfluidic channels such that they flow into these channels, so they're sucked into the channels and they get trapped at this constriction at the end, and then the individual bacterial cells are growing and they're pushing out the daughter cells to this end. And what you can do then is to apply different treatments, so different antibiotics to different portions of the cell ship. And in this case, you see that on this side, we don't have any antibiotics. This is kind of a reference channel, and you see these black sausages like this are the bacteria, and they grow rapidly when they don't get an antibiotic. But on this side, you have an antibiotic, and you may even see it by eye that it kind of grows slower. And when we quantify how fast this is, okay, so this is now, you look on individual, one of these traps for the bacteria over time. And this is the reference channel. You see that start with one E. coli cell in this case. It grows. This is over 60 minutes, so it kind of divides somewhere here. And you get multiple cells. It grows exponentially if you look at the length extension. And if you have added an antibiotic, in this case, it kind of extends linearly, which means that the actual growth rate is going down. So growth rate is the relative length extension over time. So in this case, it's a reference. It kind of plateaus for the individual cell and goes down for the treated cell. In the actual chip, we have a few hundred to a few thousands of cells, traps for each condition. And this is now what it looks like if you average the growth rate impact for, in this case, nitrofurantoin at different concentrations. You see that at high concentration, it drops quite, after 10 minutes or so, you see that you get an impact of nitrofurantoin in this case. And also a concentration dependent response, which we'll get back to. So this was promising. We did this for a number of antibiotics that we thought were relevant for urinary tract infection. And we could, so these are the kind of averages, and the dots corresponds to when we could distinguish that we had an impact of the antibiotic compared to the reference growth. This is not any resistant isolates or anything. This is just a kind of wild type E. coli 1 isolate treated with different antibiotics, and how fast we can measure growth rate response. But still encouraging is that this is time scale in minutes, and we can actually see an effect of these antibiotics in a very short time scale. But this was a very complicated assay, and to make it into the clinic and actually useful for anyone, it needed to be much more user-friendly. So it doesn't require a PhD level operator that has been trained for this for years. It also needs to be much more inexpensive. Our current standard microscopes costs hundreds of thousands of euros, and you can't have that at the point of care situation. And we still need to keep it as fast. So this is the point in 2017, we started this company to make a user-friendly version of this assay. So it should be a instrument, a reader instrument, and a cartridge, single-use cartridge. And this now actually exists. So it's being tested out on the clinics, these instruments and the cartridges. And in the cartridges, there are 5 different antibiotics, each at 5 concentrations. And then there are some reagents for making a presumptive identification, which is needed to interpret the antibiotic breakpoints correctly. So the chip is it's like the size of a CD disk approximately, so 10 centimeters. And these are the reagent reservoirs for 32 different conditions. It has a blister with the Miller-Hinton broth, and you have a port for loading the sample over here. And if you zoom in, you then start seeing the different conditions where the bacteria are treated with antibiotics. And if you zoom in even more, you start seeing these traps for the individual bacterial cells. And these are the constrictions, which are features of 200 nanometers. Yeah, I should say that please interrupt me if you wanna, if I'm not clear, if you wanna ask something. I can't see if you raise your hand, so just please shout out.
Yeah, I will make sure to see if anybody raises their hand.
Thank you, Eva. Yeah. So what happens in the analysis? So for then each of these antibiotics, we get temporal responses in growth rate for 5 different concentrations. So this is the nitro again. You see the growth rate drops fastest if you have a higher concentration. We fit this to a function, logistic function, and then use that interpolated function as and evaluate the impact after 25 minutes. So you get these kind of impacts depending on the concentration. And if we instead plot that as a function of concentration, so this was time, plot it as concentration, we can then again fit a function for the kind of concentration impact for of this antibiotic on this particular isolate that was run in the assay. So this is now one sample, and you get these kind of impacts for different concentrations. Of course, we need to calibrate these impacts for a number of isolates then with different MIC values, so different, say, resistance to the antibiotic. And that's what you see here. Different curves here corresponds to different isolates with different MIC values. And the MIC values are those that are on the color scale here. So green means it's susceptible and it responds at a low concentration. And red means resistant, and it kind of needs very high concentration to respond. So we have lots of these isolates, reference isolates, and we can determine then at what concentration should we evaluate the response and see which is the kind of impact that can distinguish the resistant from the susceptible isolates. Questions?
No, there is nothing here.
No. Okay. So then we have one second. Then we have tested this on a number of different isolates to calibrate the system and the breakpoints. So we have lots of, in this case, 260 different isolates that have been done with different MIC values. And this is in the calibration run. So to determine these kind of thresholds, this is the reference MIC value. This is the concentration impact that we measure in the instrument. And as you can see here, if you are in these green zones, it means that we're making a good job. There are some outliers where we would make a mistake. Some of these are then errors in the reference testing, and some of them are errors in the actual assay. But in general, there's a very good agreement, and we can make a classification of resistance and susceptible isolates. So this is Cipro, Nitro, Amoclav, and Trimethoprim. Trimethoprim makes exceptionally good separations always. Okay, that's that. And I should show you a video that this thing actually works and how it works. So you load a sample. This would be a urine sample, a fresh urine sample, into the cartridge. You have the instrument. It's a bit slow. You go into patient mode. You scan a barcode. Insert it. Wait 30 minutes for gram negative, 45 minutes for gram positive. And then you, if it's a positive sample, you get a response with the resistance spectra, S and R, SIR. And if it's negative, you should get that response in 15 minutes. Okay, that's it. So that was what we have been doing in the company. What we are doing now at the lab at university is taking this one step further so that we can do SpeciesID also in the test, and also mixed infections. And in this case, so if you have different species in the sample. So in this case, you would have a sample now with different bacteria inside. And same as before, you have references that are not treated with antibiotic. You have the treated sample. And then you, the different thing after that you have gotten your phenotypic response, you go in with fish probes and determine the species by having fluorescent probes that bind to the ribosomal RNA. And that means that you can then stratify the phenotypic response in a channel based on which species it was in the end. So you can pick out all the ones that have the same fish probe corresponding to one species, and look at the phenotypic responses only for those ones, so that you can distinguish. In this case, 2 of the species are responding to the antibiotic and the 3rd one is not. And this is a story that we are trying to publish now, so work by Vinod and Pranit in my lab. And if we go into a bit more of the examples here, so this would be the reference channel. Now it's 4 species that are mixed, and of course it would be very uncommon that that we have 4 species at the same time. But just to prove that it kind of works also in this case, this is the reference, and once it has grown for, I think we do 60 minutes now, we go in with the fish probes and pick out which species is which. In this case, 1 color for each species. And if we then treat with an antibiotic, in this case vancomycin, you can see that it's in this case only the enterococci that dies, stops growing. So the gram positive stops growing. The ones that are green here are enterococci, and they're not growing up here. And we can then do the same for different antibiotics that have different profiles of targeting different bacteria. And if you would have a resistant isolate, you would see it as we saw before, that it would not respond to the antibiotic. So this is 4 colors and 4 species. And to scale that up, we've also, now I should show these results first. So this is now individual experiments with 4 mixed species. And you can see with ciprofloxacin, you don't get the response for Pseudomonas. Gentamicin, you don't get the response for Fecalis. Nitro, Pseudomonas is also not responding. And for vancomycin, you only kill the enterococci. Should I say more here? No. So if we have more species, what we can do is combinatorial probes. So we have adapter sequences that bind to the ribosomal RNA, and these adapter sequences have different kind of arms. And if we have, which means that we can probe each species with 2 different colors in this case. So in this case, E. coli, it binds green and blue, Klebsiella, blue and purple. And with these combinatorial colors, you can go up to 10 species in one round. And then if you do multiple rounds, you can do even more species, of course. And it looks like this. If you load the whole chip with E. coli, you would get the color in 2 different channels. So Cy3 and 488. And with Klebsiella, you get the cyan and the purple color, for instance. And if you, so if you have a mixed sample now with 4 species, and you can then see, for instance, that this is an E. coli because it's shines in both 488 and in Cy3, but not in the other channels. I think that was it. So, in summary, we find that we can use rapid AST to guide prescription, so we can make a 30-minute test, and that should make it possible then to use old and inexpensive antibiotics, even when the resistance levels go over 20%. Since we are, so we use phenotypic AST, we look at the growth of the cell, and since we have single cells, we get extremely high sensitivity, and we get speed because we can look at the length extension of the individual cell at the single generation time. So not waiting for an individual cell to grow to 10 million cells to see it as a coloring on a plate. And in the company, the first product will be a 30-minute point of care AST for urinary tract infection. And what we are doing next in a SSF-funded project is to do in situ genotyping after the phenotypic AST, so that we can do rapid SpeciesID, and which is then also possible to use for mixed samples. This obviously involves lots and lots of people. So technology depends on things that we have developed over many years in the lab, funded by different sources. The main people involved in the AST projects are Praneet Ossi, who was in my lab before, and Vinud. And then there are, of course, lots of people at the company that are working on making this first product available to everyone. Thank you.
Thank you so much, Johan. We open now the questions and answers. Let's see if anybody raises their hand. Meanwhile, I do have maybe some questions or comments or for you. Maybe some of those are also more like curiosity. How is it you've been involved in setting up Astrego from the very beginning, from the idea of being a research project and this could be a product? How has that been? How does the journey has been so far?
I mean, it's been super interesting to see the kind of other side. Many challenges are the same, I think, as in academia. Some challenges are different. It was all very new to me, so it's been exciting all the time to see the other things, and especially the how powerful it is if many people work exactly, I mean, to solve a common goal, very specific goal. In the university setting, we typically have much smaller I mean, project teams, so maybe a few people that work on with very kind of flexible goals, whereas in the company it's so very focused with lots of people. So that's a difference.
Yeah, I mean, it's just, because we want to inspire, the new PhD students as well, that the possibility of perhaps bringing research into a company setting or developing a product, it is possible, and there is support, and there is a way to do this within from the university. And the fact that you are still, involved in both things, it says a lot too. So that's great. We have some questions actually from Philip here. He says, great talk. The applied sample to the AST device is a culture isolate from a patient or patient fluids with infected material?
So in the calibration, we have used spiked samples from clinical isolates. In the actual usage, right, or in the clinical validation, it will be a primary direct urine sample. Mm-hmm. There is a 10 micron filter built into the device to get rid of some particles.
Uh-huh. But is there going to be some step of like pulling the material, or just the pure urine as it comes? Like maybe
urine directly as it comes.
Okay. That's cool.
And the white blood cells typically don't. I mean, they flow past the small channels, and red blood cells actually squeeze through the constriction. Mm-hmm. There is blood in the urine. Yeah. So directly from urine.
That will cut the times for sure, and also the procedure.
And it's important that it's a fresh sample, so that they are
has to be in the moment.
They have to be rapidly growing essentially when you start. Otherwise, you don't want to have a lag phase in the beginning.
How is that ensured? How is there a knowledge of what stages the bacteria that are in the bladder and come through with the sample are? In which stages of the growth are?
So at least E. coli in the bladder is rapidly growing. There are some recent papers where they do Illumina sequencing of the chromosomal DNA and see the ratio of origins to termini, and see that they have like this increased fraction of origins corresponding to a growth rate of 20-30 minutes for the E. coli at least.
Right.
So, but they will depend on the species. Yeah.
Mm-hmm. Philip is also asking, how long does the calibration take? Do POC institutions, point of care institutions, get pre-calibrated instruments? Do they have to calibrate it themselves, or depending on the most common detected species?
No, it's pre-calibrated. So this is something that we have been doing with reference isolates from UCAST and from. So the calibration is made. I mean, it's a fixed calibration that doesn't change over time. So we calibrated it on a few hundred isolates, and then we have now in the validation run it on several hundred more new fresh clinical isolates, and it gives the same result, which is reassuring. For the individual clinic, the important thing is that the measurement is to a reference, internal reference, right? So you have the untreated bacteria as an internal reference, and mm-hmm. It's in relation to the reference that you measure the impact.
Mm-hmm. And his last question is, does the device determine MIC, or is it only qualitative for a given therapeutic concentration?
So it gives a qualitative SIR response. Internally, it has an MIC value, but it's not reported to the user. Mm-hmm. So it's an SIR, and should I say more about that? Yeah, we think that's the most useful for the doctor to guide treatment.
Mm-hmm. And we have also a question by Brigitta asking also, how is the presumptive ID made with the 30-minute AST instrument?
So we have reagents, and that so in like very classical microbiology, if you can grow selectively on certain media, we use the same kind of principle that if we have certain media additives, we can then distinguish the growth rates based on the species. But it's mainly gram positives and gram negatives that we distinguish, and then some sub differentiation within the gram positives because they have different breakpoints. So we need to see what is they need to be calibrated in different ways depending on which species it is to give the right SIR response.
I will follow up on this question before I relay to you the last question that we got in the chat, because I was thinking, and this is not a criticism or anything, I don't mean it like that, but what is the benefit of also having an ID, like a species identification, on top of the already given AST? Because I feel like if you just know what to treat it with, you don't really need to know what species it is so much. So what is the other value of?
So that's totally true, and we don't give the ID as a result. It's just needed to be able to give the right AST, because the breakpoints are different for different species.
Of course, yeah, that makes sense. Thank you so much for the clarification. All right, then we have Florian asking us, saying your machine is looking at growth rate. One problematic phenotype in the clinic is also persistence or tolerance. We talked about it recently. Is there any way to differentiate between cells that are die and cells that are just persisting?
Oh, not in this time scale, I would say. Also, persistence is at a lower level. Number of the frequency would be lower. Typically, we look at up to 1,000 cells, bacteria per condition, and I think the persistence levels are less. I may be wrong here, but I think it's less than 10 to the 1 in 10 to the 5 or something like that, and it would be hard to find. We have made another chip in the lab where we have 100,000 channels, and that's aimed for that particular application.
Oh yeah, so you're looking for persistence and trying to find the percentages and the prevalence in the populations through it.
Yeah, and also pick them out with an optical tweezer and sequencing them.
All right, Florian says thank you. Yeah, so I think is there any other questions? This was a shorter talk, but it also there's a lot of things that they pertain to the company as well. So I was just maybe if you can perhaps comment a little bit on how do you see the future this developing, like do you see that the machine can be developed for other type of samples? Do you see that it can become really a widespread machine that every emergency doctor's office will have one? One are your hopes, but what do you see happening?
So I think the measurement technique itself to measure rapid AST using growth of individual cells is very powerful and will be used in, I would like to see it replace all plate cultures, essentially, like to be a gold standard for how to measure growth rate response, because it's very, very reproducible with the fluidics. This particular machine for the point of care, I think urine is a good target for more of a central lab. The obvious candidates is sepsis, of course, first directly from positive blood cultures, but ideally directly from blood. And we have also preliminary data on mycobacteria. So TB would be very important, I think, as a rapid or rapid in TB cases would be in a day, right, instead of a month or something like that. So that would be, and we can see growth impacts with smegmatis in, I think, 6 to 8 hours, which is then quite good in terms of if we could make a TB assay in 24 hours, I think that would be very good.
Do the fact that some bacteria have different sizes and shapes makes an impact in the chip, and if you have to develop different kind of chips?
Yeah, you can't squeeze a yeast cell into this, for instance, right? But we have other chips where we can grow yeast cells, and I think one could have different sizes of these traps to accommodate different kinds of cells. For the urine pathogens, they all squeeze into these channels, which is good, and
it's good.
That's also, why we have started with uncomplicated UTIs where we have a kind of narrow spectrum of pathogens compared to sepsis, which is a very, very broad, it can be anything, right?
Mm-hmm. Mm-hmm. Great. Thank you so much, Johan. I don't see anybody else. Oh, Philip has another question. What have you shown so far is focused on designing the chip. Does Ostrego also do research on the chemistry of the materials?
So the material of, they're not making new materials, but it has been a lot of refinement of which material to use to make a kind of inexpensive and reproducible process for making the cartridge. They're not making any new materials, but there is of course chemistry in the formulation of the dried in antibiotics in the ship, things like that.
Mm-hmm. And Brigitta is asking, do you have a timeline for when this might be commercially available? Thank you for the talk.
So this year is the
2022
answer. Yes.
The year. That's great. That's lovely. Is this something actually now talking about that like the hospital would be the one taking care of buying these machines and putting them in the doctor's offices, or would the doctors have also like something to say there?
That works very differently in different countries, and how the different countries recommend doctors with instruments. Our plan is to work with a big company that has the market channels, because we will not be able to reach doctors' offices.
Yeah, so you don't do the marketing, is kind of like a collaboration with other company rather than
yeah.
Great. All right, Johan, it was lovely to see how this is growing and evolving, as well as the bacteria. And thank you everyone that's been with us for this seminar. We are going to have a next seminar on the first Friday of February. And we're also going to have a podcast episode released on the beginning of February as well. So we have quite some things coming up. And also just keep checking the website, because workshops and symposia and other activities that might be available will be published there. Other than that, I thank all of you for being with us today. I wish you continue having a great day and an even better weekend. Thank you, everybody.